It's white and gray on the top of the sediment surface.
That's cool.
Okay, come on out video.
So we've been talking in the chat room about trying to figure out what these little white worms are and other
chemosynthetic little creatures.
Other little creatures here, and Jill Bork said that
yeah, I have more on that one that you're swinging over to.
She doesn't think that they've seen any sclerodinium-like sibiglinids in the Atlantic margin so far.
I'm gonna do it. I'm gonna mess with your sonar a little, see if we can get these to pop.
Copy.
Yeah.
Yeah, look at all those bubbles on the right. So we're wondering whether this is some kind of patchy emission of gases from the seafloor. And there's not a well-developed chemosynthetic community here, so it might be that this is fairly new.
Okay, I'm gonna go over top and sit down on that one near the fish, the rose fish.
Going right through the bubble stream here. And we're at 334 meters now.
Okay, come on in video. Copy.
On the left, there's another one of those white, probably bacterial patches with bubbles coming right out of it. And it's interesting that the little holes have kind of edges around them that stand up.
Copy. Go ahead, watch one. Will do.
Like a laser dot width.
I think it's pretty small, Carolyn, this white patch here. We're going to try to put lasers on in a second, but they're saying it's probably about the size of one of our laser beams, so.
Can keep coming out video.
Yeah, they are.
And if we could pan up and right before we leave here, that would be helpful too.
Just come on out a little bit more video. And then, what was that last request?
If we could pan to the right side of this, is follow this little patch.
Copy.
There was something over there that moved, so I wanted to see what that is, if possible.
Okay, full wide video. Okay, come on up.
So Amanda Demopoulos says that they've seen this white fluffy material at several other seep sites in Norfolk and Baltimore seeps and just northeast seeps, but nobody's quite sure what it is yet. Yeah, and the thing that's moving is to the right.
farther to the right watch lead?
Yeah,
Yes.
well, we can see it. It's on the right side of the view here.
Oh,
See
okay.
it kind of wiggling there.
There are phosphorites that are associated with the Gulf Stream that are probably economically of interest, but they're very hard to collect because they are so solid and you can't dig them up easily. They're usually in platforms.
Okay, you want to come on in video?
Yeah, Copilot, could you shut off the laces?
So solid. On this expedition,
Actually, it's not going to bother us because I'm going right past them.
we've seen the phosphorites, we did see the manganese, but otherwise, most of what we've seen geologically are compacted, slightly liquefied.
What is that?
I think you got it. It's a fish head. It's
It's
a
a
mostly consumed fish and just the head
fish
left.
head. Huh. And then we have a different type of worm on the left there.
Wow, that's gruesome.
But they are not really
Yeah.
economic value. The potential for economic value, of course, would be these gas areas. So that's
That could be the current that's moving it.
we know
I guess this,
That'll keep them going for another week.
Yeah.
they have found.
I guess the bubble plumes don't deter
Okay.
the blackbelly rosefish from eating.
Interesting. That's cool.
Wow, that fish head looked like it was about as big as the blackbelly rosefish's head is. It was a very big bite. And you can see some of those little white worm tubes on top of him now.
He's a funny fish.
And I'm not sure if that was a Echiuran spoonworm that we saw a little bit ago, next to the worm head or the fish head.
All right.
Okay, is video clear?
Not fish.
Is that video clear? Watch lead, is that what you wanted on this site? Okay, I guess that's it.
Yeah. They're a little distracted with live events.
Copy.
Yeah, you can see your map cams, like 2 or 3 of them all around you. It's pretty cool.
So it's right around here, I thought.
Yeah, so that's that one that you were looking at before coming up in your eyes now.
Oh, okay.
But there's that. That's probably got bubbles associated. And then we saw something out over this way that might be bubbles.
Oh wow, there goes that fish again. I never did get a good look at him.
There's a good...
We'll try and get him next time, watch lead.
Roger that, he swims away every time, so it's not easy.
I got the Zeus back, co-pilot.
Okay, so we're just finished up a live event here on the ship, so hopefully there won't be as much distraction.
Momentary step in here with you pilots.
Welcome, Roland.
Starting to get some shadows in the sonar out there. 15 meters or so.
Big bubbles below you.
Go ahead, watch one. Absolutely.
Yeah, they're on.
Okay, you can come in
Watch
on a
it nav. Just a note,
partial.
when we zoom in tight, we lose them off the bottom of the, you know, they're not.
Right, yeah, we're going in tighter than they are, yeah.
Exactly. So that's.
Roger that, thanks. Wow, this is good clear water here. You can get a
yeah,
better
you
view
can
of all
grab it.
these little tiny tubes.
Have you guys tried to see what it looks like
And
when you
Leslie,
get
you
close
just
and
missed
turn on the
a
extra
fish
lights?
head that was
We
laying
haven't
on
tried
one of
the
these
extra
bubbles,
lights
and it
yet.
was moving around.
Oh my
Right now.
gosh.
We zoomed in on it, and then a black belly
Yeah, you wanna-
rosefish came
yeah.
in and ate
I
the
don't know
whole
if
thing.
we're close
Oh my
enough.
gosh.
Just sucked it all in.
Wow.
Oh, yeah.
See some little burps coming out of the...
It's
Yeah.
like it's just a little out of the pool. I can kind of inch up a little bit.
That's pretty good.
That's great.
Okay.
Yeah, it was really hard
I was trying
doing
to
the
do to see
live
if it
event,
caused
seeing
any
this on
extra shadowing to help.
outside of my
bring the bubbles
view.
out
I wanted
more.
to look.
We brought out a lot of color on the mat here. Yeah,
Oh, sorry.
blues and stuff. It's kind of nice, the bringing the red out of the sediment shows how blue the bacteria really is.
Bubbles stop.
Did it?
Did.
Oh,
Okay, video swapping back out.
Copy.
okay.
You want to go full wide?
Yes, please. Okay, so what's the sonar suggesting, F? Do we mind turning off the light bars, co-pilot?
So have you been hopping from one to the other where you
At this
see
point, there is
the bacterial mats?
Yeah, we've been
shadows pretty much
zooming
dead
in on
ahead of
each
you right
one,
now.
and I
Okay.
think every one of them has
But
had
they're
at least
about,
some bubbles coming from it.
15 meters or so out, so you can start moseying that way and we can get a move in. Actually,
Okay.
you've got a lot of
I
filter
even
right
missed
now,
the
too, so
sediment
you
collection.
can get there.
How did that go?
It went well, so
It all settles out. But you can also move the ship.
yeah. And
But
so
yeah,
these
see these-
little tiny
See
white
these
worms
dark spots? This is what I saw before.
might be
I think, or maybe there's
sclerolinum.
something different, but these dark spots with shadows near the top here, which mouse?
Small,
This
wiry,
one,
deep
these
sea
guys.
tube worms,
So I've got it straight ahead of you now at about
and they are chemo. They have endosymbionts that are chemosynthetic bacteria.
16 meters. 16 to 17 meters.
Oh, the
So you
bubbles
could
came up right
shot
next
there. Where
to
did you go?
the black belly rosefish.
They don't seem
17
bothered by
straight
this
No,
at
ahead.
all.
I wonder if they like it.
He's about
Look at that, right next to him.
there-ish.
Okay, I'm going to sit down and try and get a shot of that real quick. Copy.
That one's an active. That's a little more active than the previous one, isn't it? Have you seen him with that many bubbles?
Okay, you can go in video.
There have been a few now that have been more frequent bubbles than the first things we saw. Yeah.
So yeah, so I dropped a couple out here for you. These two here. Both
Okay.
sort of line up and they've been giving you
If we can get a
Do you want
closer
to show me
look,
a video?
we can see if there are any more of those little white tubes.
Co-pilot, drop the lasers, please.
forget to turn them back on before.
Okay, it's not the white tubes that are the sclerolinum, but they're associated with those in the Gulf of Mexico.
It's interesting. Must be like collecting under there. Once it reaches a certain size bubble down there, like, let's go.
You scared it.
Copy.
Do we know what that little bauble is on the right? It looks like a miniature sponge, but I doubt that's what it is. Maybe if we can zoom in on that a little bit.
We saw something that looked like an acorn worm in the other patch. I wonder if this could be one of those that's mostly retracted.
Okay, why don't we
And
go
those
full wide
tubes
real
are
quick?
plentiful.
Okay, now you can go back in. Okay, why don't you come back out a little bit so we can find out where it is?
Yeah, I think it's down below.
Bob Carney suggested that these squiggly white
Okay, hold
white
it there.
tubes could just be another large microbe morphology.
Yeah.
I think we lost it.
Okay, if we see anything that looks like a ramen noodle, that might actually be the sclerolinin.
Ramen.
No, I think we'll move. Okay, full wide. And then lasers on. Gonna take the rotator real quick.
So it's interesting that this seabed isn't as pitted as what we've been seeing elsewhere. We're seeing the little holes, but not the larger burrow-like holes.
Sure. That sounds, yeah. Yep. Okay, you can go past
When
this
we
kid's video.
took the sediment sample,
Oh, that's
could
a
you
pretty
tell
big
if we
seep.
penetrated into the darker layer at all?
It didn't look
Are you
that
just
much
past
like
the
that
skids
to
video,
me,
or
but
is you
I
going a
actually
little harsher?
was having a hard
Okay.
time concentrating on
Yeah,
any
I'm
one thing that
sure.
was going on.
We were very distracting. I apologize.
No, it's not your
Nothing we
Yeah,
could do
we-
about it.
Yeah. I wonder if that's another one of those southern
you've got
hakes in
some
the distance
Big
there.
targets coming.
Okay, so should we kind of go out and see what they are?
Yeah.
Okay. What's the bearing?
Straight ahead. You can see them on your sonar, Chase. Those
Ah,
shadows.
gotcha.
See the sort of yellowish red with shadows?
Yep.
Hoping those are largish seed targets or something.
So we've got some targets on the sonar watch leads that look like some larger seeps.
Great. We can just take a quick zoom here and keep on trucking then.
Okay. Quick zoom at this mat over to the left of the lasers, or something else.
Yeah, this mat right here, just to
Okay.
the left of the lasers. And if you all didn't hear that,
Okay, come on in.
it sounds
video.
like the pilot sees some other target in the sonar up ahead. So we'll look at this and then keep
Yep.
on getting it.
Yeah, there's something peeping out there.
There's another one of those,
That's what you saw before?
yeah, possibly acorn worms or... Yeah, did you see what Bob Carney said about the scoop sample?
Yes, yeah.
I think we're too far away right now. Is that full zoom video, or?
Yes, it was.
Okay.
I didn't see anything coming out of this one.
No. Didn't either.
just past the skid.
Thank you.
And we might have seen another proboscis of an acorn worm sticking out there.
Okay, so the sonar is showing it should be a bit upslope. Kind of looks like there's seeps all around here, Nav.
It's hard to tell which botches are. Yeah, we sort of lost those. I dropped a target. You can see here on high pack.
Okay.
The last one that we were looking at, that's where roughly it was.
Okay.
So just aim that direction again. See if those dark spots come back. Those dark shadows come back, or if it was just luck of the way we were looking, or what it was. It's like. Do you want a ship move?
Copy that.
Which direction?
It works for me.
Okay. Yeah, 245 is more on track anyways. Yeah. Should be looking right at it now. I don't see. Well, I guess I see a couple of dark spots there, but it's to the left. But might have just been bogus.
Okay.
It's still like 5 or 10 meters away from you. But go ahead, watch one. Oh, on the right, no, those are just holidays, sonar holidays. Those like swirls. Yeah, that's Sirius. And
Turning.
yeah, those are just little holidays. Yep. Bridge nav. Yeah, can we do a ship move, please?
Copy.
We'll do 20 meters, bearing 245 degrees, speed .2 knots. That's affirmative. Thank you, Bridge. I guess if we turn the corner for this waypoint 4 here, so follow the uphill. He's going to come over you.
Okay.
And then about 20 meters. He'll end up around here, and then we can go pretty much south for the rest of the time to get to 4 and then eventually 5.
Okay.
We have roughly 10 minutes left in this watch.
Copy.
We have a little more than an hour and a half left of this dive. We're still climbing. Our destination is the peak of this ridge that we have been traversing since a depth of 510 meters. It is one of the intra-canyon ridges of the Hatteras Canyon. And it's been at times extremely steep, especially on each of the flanks. We're riding up the backbone.
And we've seen this sediment cover throughout the dive.
Seen
But
a few.
It has varied
pop up.
considerably in
Tilt up.
the seascape at the base. Oh, look at all those squid. Oh, my gosh.
That's beautiful.
Oh, my gosh.
This is probably more on the line of what Mike Vecchione was talking about with the
I hope he's watching.
larger schools of squid.
Off into the universe. Look at them all in the distance.
Like shooting stars.
Yeah, like shooting stars. That's funny.
That was beautiful.
Squid shower.
We haven't seen too many of their predators here.
About to touch bottom.
Copy.
The wreckfish would love this habitat.
Yeah.
We saw several wreckfish, each of which was close to a meter. Some of them were larger than a meter in length.
Looks like the ship's caught
And
up.
they had found some wonderful habitat on the Lophelia Banks Deepwater Marine Protected Area off of Cape Fear. I guess it's known as the Cape Fear
Then we can start you guys
Lophelia Banks
Copy.
Deepwater Marine Protected Area. I haven't seen anything quite that large here. But as I was saying, the beginning of the dive,
I think a lot
the
of
base
those
of this
shadows
steep
are
slope,
maybe these depressions
we
here.
saw a lot
Yeah,
of
could be.
small burrow-like pits. Many, many small fish
Pooled.
of two varieties of this
Thank you, Bridge.
black belly rosefish that you
The ship
see
move.
just to the right of the lasers.
Yeah.
And the eel pouts,
Nice line
which
up.
you see on the left of the
Next to the
lasers.
wheel.
And we saw a lot of juveniles there.
Coming back.
And then as we began to climb, interrupted again by the squid.
They're so awesome
sorry,
to look at.
Watch.
They are. And they're not being disrupted by us. There's no ink following them, leaving the water pretty clear.
Oh, yeah. So that's the Elex squid that we've just been watching. And these look like larger burrows.
Yeah.
We've been chatting in the chat room about whether any of these may be made by tilefish.
Okay. 180 copy.
So maybe right now the tilefish are near the surface feeding.
Swing around. Well, let's take a look at this real quick.
I don't know if they feed at the surface, but we certainly haven't seen them here.
I know, we haven't seen them at all.
I know they're bottom dwellers.
It seems that if these were made by tilefish, we would have seen at least one by now,
Yeah.
unless we scare them away.
And
with
they're
our-
so deep. I wonder if they make them that deep.
Yeah, it
Try
sounded
and
like
fly over this one and look
I
down.
think it was Andrea Quattrini who said that the burrows are wide and then straight down.
Oh, okay.
So these
Yeah. well,
don't look that wide. Oh,
what's interesting, though, is that there are no mounds of sediment associated with these. These are just holes, which makes me think they were excavated from underneath by something, and the sediment fell into a cavity, like maybe more from gas. And that's very undisturbed sediment around the edges.
yeah. And we've been noticing that it's not typical of some kind of crab or fish making this hole.
Although maybe from the side view, this view doesn't look like there's a big mound, but it might actually be from the side view. But regardless, it wasn't excavated very recently because it's fairly undisturbed.
Definitely looks deep. When there's a, is there a black-bellied rosefish in the bottom of that one, maybe?
Oh, way down in?
Mm-hmm. Or maybe, no, it's not the laser.
I think it is reflecting off the particulates. Yeah, that is the laser. It's just the beam of it, not the dot of it.
What kind of creature made that mark?
Something big.
John Reed commented that if we look into the burrow, it should have a shaft at the bottom where whoever created it would be sitting. I don't think we've seen any shafts, and that one looked like it kept on going.
So the 6.8 looks like it has a full turn.
Yeah, almost.
We kind of want to start heading south, right, Nev?
Yeah, whenever you guys are lined up for that.
I think I'm lined up on the local high right now, pretty much.
Okay. You want a southerly move? Or you want to push out a little first?
Yeah, I guess I'll push out first.
Either way. Whatever your preference is. Well, whatever's good for you guys uphill. Doesn't have to be due south. I just picked south as a general direction.
On D2 sonar, it looks like 210 or so is upslope.
These submarine canyon areas are affected by a multitude of forces of Mother Nature, from the sort of the
What'd you guys line up on? 200, 210?
overpowering constant Gulf Stream, and even though the Gulf Stream is constant, its axis varies in its position. It moves inshore and offshore, so that any one location on the seafloor may or may not be impacted from day to day by it, or varying degrees of influence. But in some of these areas, even, oops, I think we're just passing over a bubble.
Coming out of the dark spot.
The dark spot to the left and a white spot up front.
Yeah. But even tidal influences, maybe not at this depth so much, but farther up the canyon where it's shallow, tidal influence cause
ship move, please.
Changes in current direction and flow and strength.
Degrees, speed decimal
and
2 knots.
Major storms that come through this area, especially this very narrow shelf area, have some impact on the heads of these canyons. And all of those factors contribute to moving sediments around. And at the heads of the canyons, a lot of those sediments get channelized into the canyon itself, and they stream down and help to further erode the bases of these channels. And where we are located is on one of the ridges between some of those channels. And we're not on the main axis necessarily, but they erode away at the base, and the upper portions of the walls of the canyon begin to tumble down through time.
Changing over.
And erode deeper and deeper into the underlying continental shelf.
This looks very similar to the last ones that we looked at, these patches of white bacterial mats. And see if there are any bubbles coming up from any of these small holes.
We have a watch change on the front row. John Reed, in reference to the tilefish burrows, he said earlier that they sit in their shafts looking out for prey that swim by, just like the jawfish. They're looking for the jawfish. I don't know what a jawfish looks like, but I suspect
Yeah,
there's a large jaw.
All right, video, come on.
Yeah, pilot, we got a... That gray vent on the right side center has been bubbling.
Okay. Oh.
There's a bunch more in the area.
Understood.
Looking at you at 70 degrees, pilot.
All right. Must be these dark spots with white next to them. Probably.
yeah, if we can take a look at this one, pilot, that would be great.
Yeah.
Okay, we got vent probably right underneath the right laser.
Okay. You can frame it up.
Mm-hmm.
Oh, okay, we had one to the right. There's another one.
There's some bubbling on the left.
Oh, yeah.
And
Left,
right.
Yeah.
right center.
Well, the bacterial
too
mats
active.
give it away, don't they?
Okay, that is 90 degrees,
Yeah, and it's
pilot.
particularly white where those bubbles just came out.
And the black belly rosefish just sits in the middle all of it. At
And
least
the big
here.
one.
There's some more on the right.
Yeah, that one on the right is popping. Was just popping.
You could
There
come
aren't too
in.
many other organisms, though,
No.
around this little landscape.
Looks like we haven't fired any of the Niskins yet.
Any idea what that is?
We've been seeing that, and Amanda Demopoulos said that they've seen this in the mid-Atlantic seeps as well, Baltimore and Norfolk, and then all the way up through the New England canyons, but not sure what it is yet. It looks like marshmallows.
All right, video. Pilot's clear. One video is clear. All right, now looks like we're headed kind of south. Is there a waypoint 5 or not?
Yeah, we're heading south.
Okay.
Thank you, Bridge. Go ahead, watch one. We are about 5 meters from
We
waypoint
have
4.
another Tina-4 passing our view with its rows of cilia that propel it. It's also known as a comb jelly.
Yeah, we have a plume out of this mat.
And it looks like more bubbles and
Oh, yeah.
another little white patch.
Maybe you can frame this one up. Oh, there it is. Got it.
There's tons of squid above the D2 if you look at the Sirius view.
Oh my gosh, I hadn't noticed. I wonder if they like bubbles.
My bubbles.
It would be nice if they did, because then we could just use our sonar to find the squid patches.
Yeah.
It'd be easier than finding bubbles.
All right. Video, you can come. Oh, I guess quite a bit of dust blowing through here. Pretty turbid water.
Again, those little holes that have kind of edges, they're built up a little bit. We haven't seen anything coming out of them, though.
It almost looks like they have some sort of interior lining.
Thanks,
Is
video.
that? Have we been able to zoom in on one real close?
I can
Fairly
get
close, but
I can get closer. Can take a. All right, video, come in.
it's another one of those white marshmallows.
They're
Or whatever they
behind
are.
the seat towards the
They're certainly not marshmallows.
Yeah, they are.
There they are.
Yeah, that's what I'm looking at. Thanks, pilot. Yeah, I was thinking the one right at the top center screen looked like it had something, but I think it's just the bacteria. Yeah, several of those have a little bit of white at the lip. Thanks very much.
Just stir it up for us.
All right.
It
Pilot's
sure would be
clear.
interesting if those are organisms that are liking these environments. So we didn't see those pits lower on the slope. We saw them a little before we started seeing the bubbles.
Right. And Scott France suggested that the white marshmallowy looking thing we're just seeing might be a bacterial film that's trapping the bubbles. And it does look like that because whenever a bubble comes up, it moves.
We just went right there. It is.
This is quite an active one here.
Yeah, there's two of them.
Okay.
Just tilt down.
Yeah. Come in if you want
Okay.
it. Where'd it go?
It's right
Oh,
there.
there it goes.
There's one to
Yep,
the
and
right,
another
one below
little white.
us.
That's a great bubble stream there.
Yeah.
that's a good one.
These fish keep stirring up sediment for us.
And Scott France said, "Old Faithful in miniature."
Just thinking that. All right, video, come wide. Too much sediment in this area. Blowing around.
Looks like there's a larger
Ooh, look at that one.
burrow type thing up ahead here. Yeah, and the bubbles aren't totally continuous.
You saw at least 4 distinct places go off there in that little patch.
Sorry, Carolyn, could you repeat that?
And so that was really cool a while ago. We just saw 4 distinct places go off in that little patch there.
Uh-huh.
That emitted bubbles.
Come in if you wanted. See if there's anything down here. This might be abandoned.
And just, I wasn't on the pre-dive call, but of course you guys imaged something in the water column. Obviously, all these little things we're seeing go off, they're so close together that they would effectively look probably like one plume in the water
Yeah,
column.
but you come out. Maybe there's a different one that's got
Okay, yeah.
something in it.
I was wondering because they are pretty close together.
Well, you can get resolution sub-meter sometimes, but, sometimes you can't. So it is hard. Sometimes it just looks like they all merge into one big plume, essentially.
We're now at 330 meters. But this is good ground truthing of that. You know, we're able to see this bubble plume when you went over it, Carolyn, a few years ago, and then we saw it again today when we passed over it with the multi-beam.
Okay, video, come in.
Now we know what it looks like at the seafloor.
I'm still hoping for a major venting here somewhere.
And maybe a full chemo community would be
That's right.
really great.
So many squid. Look at them all. It's crazy. Yeah, but you can come wide. Keep moving.
So, Cheryl, I'm sorry, since I wasn't on the pre-dive call, I don't know whether Derek found any sort of hard bottom based on the backscatter last night, or if you had other indicators that there could be any hard bottoms here.
I don't know if I don't remember what Derek said about the backscatter.
I think his impression was that this whole area would be sedimented as we're seeing it, unless there were steep exposures, which you can't tell on our multi-beam, depending on the direction you're flying. But
Yep.
yeah, but the backscatter, I think, was pretty consistent along the ridge. So I think what we're seeing is what we're going to get. But he's not here right now, and I just saw it briefly, and we didn't really discuss it that much. I don't know if we talked about the backscatter at all, actually, on the pre-dive call.
Come in if you wanted the video. What's that small white thing?
Fish head.
Ah.
Not sure what that is.
All right, video. Come wide. We'll keep looking. See what else we see here.
And the big well, what's dropping right now is a fish head, and this guy may gulp it.
Oh, yeah.
There's a question in the chat room from Jim Masterson. This is Carolyn Rupel with the U.S. Geological Survey. It regards what was causing the bubbles that we saw coming from the seafloor. So this entire area, as is much of the Atlantic margin at these water depths and down to say more than 1,000 in some places, 1,000 meters in some places, there's a lot of gas down there. And the gas is generally generated in situ, in place, in the sediment by microbes that feed on organic carbon that's deposited in the seafloor sediment. And so the methane is generated there, or sometimes it's older microbial methane that's migrated. So all of these sort of shallow areas, like we're looking in the relatively shallow, about 300 and some meters right now, are very full of gas just underneath the surface. And in some places, that gas is actually going to make it to the seafloor and vent out.
Cream?
And the story on this margin, just briefly, is that before about 2011, there were only about 3 or 4 places in the entire margin that were identified as leaking methane gas. And we now know, based on some work that was done by NOAA Ocean Exploration, U.S. Geological Survey, and others, and published in 2014, that there were over 500 places leaking methane. And then subsequently, the USGS has done about 5 cruises, and then we found hundreds of other steep locations. So the whole margin is leaking methane, and in various ways, sometimes associated with really nice chemosynthetic communities at the surface, and other times just sort of leaking from bare seafloor. So I hope that answers the question. If not, Jim, feel free to put another question back into the chat room.
Thank you for that great answer, Carolyn. And thank you for asking it, Jim.
Yeah, Jim has a group of high school students at Harbor Branch Oceanographic Institute, and we say hello to them. And we're glad you are joining us. And for your information, we have about 30 other scientists online, so fire away with questions. We've got someone here to answer them.
This is video editor Emily Narrow, and I have a question about all the fish heads that we're seeing on the seafloor, and I was wondering if that's a behavior of the squid above us. There's a massive school of squid above us, and if they're eating the fish and just not eating the heads.
I wonder if Mike Vecchione might be able to answer that. I mean, it's a pretty likely hypothesis. I haven't experienced this myself, so... Mike Vecchione, are you within telephone reach right now, or?
That's a great question, too.
It is a great question, because we really haven't seen the fish heads on the seafloor until today.
We've seen a fish take one, right, off the bottom. But that would be a good hypothesis.
Crabs over here.
And here's another hypothesis. The march of the cancer crabs.
Is waypoint 5 the last waypoint?
Yes.
Copy.
I wonder if Stephanie knows about that.
Yeah.
She might.
Yeah, she's not with us at this moment, but we'll keep that question in mind, Emily, since we can't answer it yet. We know where to find you.
Coming partial on crab.
Go ahead.
Hey, Mike Vecchione here.
Sure.
Oh, hi, Mike. How are you?
I'm okay. I'm calling because you asked me to.
Good.
Great. Did you hear that question?
I did hear the question, but I didn't actually see the squid heads that you were talking. I mean,
the fish heads.
The fish heads. Yeah, there was one. I don't know, maybe a half hour ago.
There might be one right there.
Right
in
here.
fact, we're going to zoom in on it. It looks like one of the what do you call them?
Barracudinas.
Barracudinas. At least it's shiny.
Come out a little bit. There's a bunch more.
The fish head was sitting there on one of these bacterial mats, and it was moving around because of the bubbles coming out of the seafloor.
Yeah, they're all over the place.
But yeah, they're all over the place here.
It's littered with.
And
come here.
Well, the short answer is it's very likely that is a result of predation from the squid.
Wow.
A lot of times when a squid grabs a fish, what it'll do is bite it right behind the head and sever the spinal cord.
Wow.
Actually, what I've seen before is the exact opposite, where they'll start eating at the head end.
Oh.
So there'll be you'll see squids swimming around with a fish tail coming out from between their arms because they're munching on the fish from the head, working their way back.
Wow.
Huh.
But sometimes they'll also basically fillet a fish when they're eating it and then throw away the spinal cord. So they're capable of doing a lot of things, but it wouldn't be surprising at all to find out that all those ilex are feeding on the fishes. I've seen several of the ilex grab fishes out from the lights as they swim by. So they're probably severing the spinal cord and I guess just dropping the head and eating the body in this case.
Wow. That's really interesting. It was a really good observation, Emily.
I wasn't paying attention to the fish heads on the bottom, but I was watching the squids grabbing fishes out of the lights.
Yeah, there's quite a few fish heads, and they're small. Pilot, I wonder if we could stop and look just behind that black-belly rosefish.
Because it looked like they were feeding on myctophids.
Yeah, I mean, I don't know what these types of...
great bubble plume, too.
Yeah, I don't know what... there have been several types of fish heads. There's about 3 here right now, and they're tiny.
That would be consistent with myctophids. And for the people that are listening on the outside, myctophids are called lantern fishes.
That was a recent.
That looks like a possible myctophid head. You're right, the jaw is still twitching on it.
Wow.
We're exploring a bunch of bubble blooms in this area right now.
I've seen myctophids swimming around in the lights. These lantern fishes are midwater fishes, but when they get in an area like this, they're vertical migrators, so they migrate down during the day and up at night.
Is this something we could do
when they get in
segments?
an area that's constrained by bottom depth, then they sort of get aggregated close to the bottom because they can't go as deep as they'd like to.
He just moved his mouth.
And that makes them good targets for anything that wants to eat them.
There's a couple more between my toes
and benthic animals in the area as well.
Wow. Well, looks like there is quite a feast going on.
Yeah, they're littered.
Yeah, you should send a frame grab to one of the fish people to get
it's a good copy.
An idea of what kind of fish that is.
It's hard to
my guess
estimate
is
how long.
a lot of these are myctophids that the squids are eating.
Okay. Hopefully Andrea Cotrini can take a look. I'm sure she would know make sure that these are myctophids for us.
That sounds good.
But look at all of them.
Wow.
Good. Oh yeah, there's a lot of them there.
And the first one that we saw, it didn't look like a myctophid to me. It looked a bit larger, but we were looking at it from the back end also, and we were way zoomed in.
Squids are not very picky when it comes to what they will grab and try and eat. They'll try and eat the sub if they can.
How much current do you have today?
Is that one in the middle of fish head?
We have
no
ATCP.
I guess it is.
I can't tell.
What's the?
Yeah. Well, thanks, Mike. That's very interesting.
Okay, that was a myctophid that just swam through the.
Okay, good.
Yeah.
Wow.
Okay. Well, I'm going to hang up now.
Thanks, Mike.
Thanks, Mike.
Bye.
And Emily Scott France said that you should change your career path to science. It's never too late.
It starts with observations. Emily, I'm sure you've learned a lot on these expeditions, haven't you?
Yes, I have. I actually majored in biology, and you'll find that a lot of the film people on the ship are film/science people, so it helps to have a foot in both camps.
No
yes,
kidding.
it sure does.
Especially because you have to document so much, not just visually, but also with your captions and the short films that you do, which are